A very good morning to all of you. Today we are going to discuss another topic in biochemistry, that is plasma proteins. प्लाज्मा प्रोटीन्स को डिस्कस करने से पहले हमें एक चीज समझनी जरूरी है कि जो ब्लड है it has two components. One is plasma and another is serum. There are a number of differences between plasma and serum. But one main difference which separates these two components, जो प्लाज्मा को सीरम से डिफरेंशिएट करता है, that is presence of fibrinogen, or you can pronounce it as fibrinogen. जो प्लाज्मा होता है इसमें ब्लड सेल्स होती है, प्रोटीन्स होते हैं, and it also includes this fibrinogen protein and clotting factors. Plasma contains all the cells including fibrinogen and clotting factors. Now what is the serum? Serum also contains these cells, proteins, excluding fibrinogen and blood clotting factors. जैसे कि मैंने पहले ही कहा कि प्लाज्मा में सेल्स भी होती हैं, प्रोटीन्स भी होते हैं और उसमें फाइब्रिनोजन भी है और क्लॉटिंग फैक्टर्स भी है, लेकिन सीरम में सारे ब्लड सेल्स होती है, लेकिन देयर इज नो प्रेजेंस ऑफ fibrinogen and clotting factors. This is the main difference which separates plasma from the serum. So we can say, in other words, plasma is a physiological fluid. Plasma is a physiological fluid of our body. While as serum is prepared in the laboratory. कब prepare करते हैं laboratory में? When you do the coagulation of the blood, there will be two components. One clear fluid which comes after this centrifugation or coagulation, that comes as the serum, which is excluding, which is deficient in the fibrinogen and clotting factors, and the plasma part that includes all these blood cells including fibrinogen and clotting factors. That's why I said कि plasma जो है, this is a physiological fluid, and serum जो है, this is prepared in the laboratory. We cannot get serum directly from the blood. We have to prepare serum from the blood. That is after centrifugation and coagulation process. Is this clear? I hope this is clear to everyone. Now, अब हमारा जो टॉपिक है, that is plasma proteins. This, as I said, plasma contains all the proteins. How will we differentiate these? How will we separate these plasma proteins from the plasma part of the blood? We need to do a process known as electrophoresis. Electrophoresis क्या होती है? On the basis of charge separation, we differentiate these plasma proteins from the blood. Here I will show a graphical representation of electrophoresis. Electrophoresis is based on charge separation. You know, there are only two main charges. There are only two charges, that is positive and negative. जब हम इस प्लाज्मा को इलेक्ट्रोफोरेसिस प्रोसेस के लिए यूटिलाइज करेंगे, there will be components bands like this. This is the major peak, that is the albumin. This is positive charge, this is negative charge. This is the second bands, here is globulins. Globulins फ्रैक्शन जो है इसमें डिफरेंट टाइप्स ऑफ प्रोटीन्स होंगे. फर्स्ट वन इज अल्फा 1 ग्लोबुलिन, सेकेंड वन इज अल्फा 2 ग्लोबुलिन, थर्ड वन इज बीटा ग्लोबुलिन, and then there will be another band that will be gamma globulins. Okay, albumin is predominant protein present in plasma. This is the most predominant protein in the plasma. 60% of the plasma proteins are composed of this albumin protein. Then I said second fraction of this plasma will be, because this albumin will be having negative charge, and that's why it will come towards the positive side of this electrophoresis pattern. और जिन प्रोटीन्स का चार्ज पॉजिटिव होगा, they will be towards the negative side of this electrophoretic pattern. That's why I said first predominant will be albumin, then there will be globulins, then there will be last one. If it is electrophoresis of plasma, then there will be another protein. I hope you will answer this question. It will be the fibrinogen, because plasma contains fibrinogen. अब अगर ये इलेक्ट्रोफोरेटिक पैटर्न सीरम का होगा, तब जाके हमें फिब्रिनोजन का पैटर्न नहीं आएगा इसमें. लेकिन दिस इज द इलेक्ट्रोफोरेटिक पैटर्न ऑफ plasma proteins. We will find this last band, that is fibrinogen band. Albumins are the predominantly present. It's the major protein present in the plasma. Then इसकी concentration होती है around 3.5 to 5 gram milligram per deciliter. Major fraction of the plasma proteins. Now we will discuss. हम अब एक एक करके हर किसी प्लाज्मा प्रोटीन को डिस्कस करेंगे. Now our first plasma protein is albumin. इस albumin protein है, you should know this. Which organ of our body synthesizes albumin? This is synthesized by liver. This point you have to remember forever. Liver synthesizes the albumin protein. यहाँ थोड़ा सा आप दिमाग लगा के ये सोचेंगे कि लिवर जो है, अगर हमें लिवर की फंक्शंस असेस करनी होगी, द कंडीशन, द स्टेट ऑफ लिवर अगर हमें असेस करनी होगी, so we will take the reference as albumin. If liver synthesizes the albumin in good proportion, then we have a good functioning liver. We have a healthy state of liver. So assessment of albumin protein in the plasma gives us an indication of the liver की जो हालत होगी, जो condition of liver होगी. If there is any deviation in the albumin concentration in our plasma, either there is decreased albumin concentration, we can directly jump to the conclusion that there is some problem with our liver. So it's a good indicator of the liver health, albumin protein. Second चीज जो albumin की है, this albumin concentration हमें we know we have a reference range of 3.5 to 5 gram per deciliter. Sometimes albumin की जो concentration है, it increases in other disease states. That is the second character of this albumin. Now we will see the functions. What are the important functions of the albumin protein? The first function of the albumin protein is maintenance of maintaining oncotic pressure. What is this? This is a very important function of albumin protein. Maintaining oncotic pressure. बेटा ये oncotic pressure क्या होता है? Actually, this oncotic pressure, it's a function of osmotic pressure. Osmotic function. वही, it's a property of osmotic pressure, this oncotic pressure. What is oncotic pressure? Oncotic pressure is the equal distribution of blood or body fluids throughout our body cells. Equal distribution of body fluids or blood throughout cells, throughout body cells. That is oncotic distribution of body fluids. Now, what is that oncotic distribution? I will tell you a simple example. We have a heart. This heart supplies arteries, vessels to different organs of the body because heart pumps the blood. Now, the vessel, then if we puncture, see, I will show it here. If there is a heart, this supplies the blood to different vessels of the body. Okay, I will use another pen here. We have the luxury of using that. This. Okay. यहाँ पे अगर सी ये हार्ट है, यहां से ब्लड पंप होता है टू डिफरेंट ऑर्गन्स ऑफ द बॉडी थ्रू आर्टरीज थ्रू दीज वेसल्स. अगर हम इस पॉइंट पे पंक्चर करेंगे एक आर्टरी को तो क्या होगा? दिमाग कहता है कि यहां पे ब्लड ज्यादा आना चाहिए, लेकिन अगर हम इस पॉइंट पे करेंगे या इस पॉइंट पे फ्रॉम द फार अवे फ्रॉम द हार्ट, इफ वी पंक्चर दिस artery or vessel, there is same amount of blood will come at that point. How is this possible? This is possible due to this oncotic pressure. Otherwise, यहाँ पे ज्यादा blood आएगा, यहाँ पे कम blood आएगा, या तो यहाँ पे toe of the feet वहां पे हम पंक्चर करेंगे, same amount of blood will come. How is this possible? This is due to the oncotic pressure. और वो oncotic pressure कौन maintain करता है? That is maintained by the albumin protein. This is the very, very important function of the albumin protein. It maintains this osmotic pressure, oncotic pressure throughout our body, throughout different organs of the body. Now, here you will see another example of this oncotic pressure. Sometimes you might be asked the clinical significance of albumin protein. The clinical significance of how this, first I will just explain a little bit more this oncotic pressure. You see here how this oncotic pressure is maintained. We have blood, we have the body cells. See, due to the presence of albumin, you should understand this. It's a very beautiful concept. You should understand this concept. There is albumin, presence of albumin in the blood, in the plasma. That means, what does that mean? उसका मतलब ये हो गया कि this is high solute concentration. Now, यहाँ पे ज्यादा solute concentration है. सेल में there is low solute concentration. होगा कि नहीं? There is more albumin in this plasma. There is less albumin in the cells. Means this is high solute concentration and this is low solute concentration. Now here comes the concept. Our blood volume का जो major component है, fluid, जो fluid part of our blood है, that's the water. Now here is more solute concentration, there is less solute concentration. There is the osmosis of diffusion. But when we talk of water, we cannot say diffusion. Diffusion means from higher concentration to lower concentration. But here we talk about the water. यहाँ हम पानी की बात कर रहे हैं. तो यहाँ पे वाटर कहाँ से कहाँ माइग्रेट होगा, बिकॉज हायर कंसंट्रेशन ऑफ सोल्यूट यहाँ पे है. दैट मीन्स देयर विल बी लेस वाटर, देयर विल बी डिक्रीज वाटर. सेल्स में लो सोल्यूट कंसंट्रेशन ऑफ एल्ब्यूमिन है, देयर विल बी मोर वाटर इन द सेल्स. और उसकी वजह से वाटर विल कम फ्रॉम हेयर टू हेयर. सेल से पानी यहां पे आ जाएगा इन द प्लाज्मा. अब यहां पे मुझे आई नो कि यहां पे कुछ स्टूडेंट्स को डाउट होगा, सर अब अगर पानी सेल से बाहर निकल के इसमें आ जाता है, दैट विल कॉज द प्लाज्मोलिसिस. बट वी हैव अ reference value, we have a fixed amount of this albumin. It's not we have 100 milligram of this albumin in the plasma. We have a 3.5 to 5 gram deciliter of this milligram per deciliter of the plasma. We have a fixed concentration. So the point is how albumin maintains this oncotic pressure, how it maintains the equal distribution of blood throughout the body. This is how this is possible. अब यहीं पे दूसरी सिचुएशन अराइज हो सकती है. वी कैन से दैट इज द क्लिनिकल सिग्निफिकेंस ऑफ एल्बुमिन प्रोटीन. यू सी हियर दिस एल्बुमिन, देयर इज अ क्लिनिकल सिग्निफिकेंस में हम कह सकते हैं व्हेन देयर इज डिक्रीज्ड एल्बुमिन, देयर इज डिक्रीज्ड एल्बुमिन, वी कॉल दैट एज hypoalbuminemia. Hypoalbuminemia. This is decreased plasma albumin concentration. अब इसकी वजह से क्या प्रॉब्लम होगी? Now same, there is decreased albumin in blood. There is decreased albumin in blood. ये hypoalbuminemia कब होगा? When there is malnutrition. When there is malnutrition, आपके डाइट में प्रोटीन्स की डेफिशिएंसी है. We take this from the diet. There is deficiency, there is malnutrition, there is nephrotic syndrome. उसमें भी hypoalbuminemia हो सकता है. अब जब hypoalbuminemia हो गया तो उसकी वजह से especially protein energy malnutrition, दो diseases main है, Kwashiorkor and marasmus. If you know it correctly, there is a difference between Kwashiorkor and marasmus. There are a number of differences, but one point differentiates from the other. In Kwashiorkor, there is the presence of edema. There is the accumulation of water in the interstitial spaces or crevices. But in marasmus, there is no edema. How that occurs? See here. अब blood में albuminemia है, blood में albumin की concentration कम होगी, means low solute concentration. और सेल्स में अब हाई सोल्यूट होगा कि नहीं? देयर इज हाई सोल्यूट कंसंट्रेशन. अब सेल्स में आप कहेंगे सर सेल्स में तो एल्ब्यूमिन नहीं होता है, तो हाई कंसंट्रेशन कैसे होगी? वहां पे भी ये इसका आंसर ये है कि सेल्स में ऑलरेडी कुछ अपने और प्रोटीन्स है और इलेक्ट्रोलाइट्स हैं. नाउ द पॉइंट इज देयर इज लो सोल्यूट कंसंट्रेशन इन एल्ब्यूमिन, देयर इज हाई सोल्यूट कंसंट्रेशन इन द सेल्स. वाटर, उसका मतलब लो सोल्यूट मींस there is increased water in the plasma. This water will move from higher concentration to lower concentration. उसको हम osmosis बोलते हैं, normally बोलते हैं diffusion. लेकिन when we talk about water, it is osmosis. So water will go from plasma into these cells, into these interstitial spaces and cause the edema. That's the problem which occurs in the Kwashiorkor. उसमें edema होता है because there is hypoalbuminemia. Water will go from plasma into the interstitial spaces between the cells and the crevices, and that will cause edema in Kwashiorkor, which is not present in the marasmus. This is one of the most important function of albumin. Apart from that, oncotic distribution, it also helps in the transport of nutrients. ट्रांसपोर्ट फंक्शन भी है albumin protein की, it transports free fatty acids, it transports cholesterol. Another very important function when it comes to this clinical significance of albumin, that is this albumin binds with bilirubin and forms albumin bilirubin complex and prevents transport of bilirubin into the, it prevents bilirubin to cross the blood brain barrier. You know that brain is that part of our body which is highly sterile. There cannot be anything which comes and goes into the brain. This albumin prevents bilirubin to cross the blood brain barrier. If that can cause, if that can cross that blood brain barrier, इसके बहुत सारे repercussions हो सकते हैं. So this is another function of albumin protein. We can say that's the most important clinical significance. It prevents bilirubin to cross the blood brain barrier. Apart from that, सबसे ज़्यादा जो important function albumin की है, that is the osmotic function or oncotic development of oncotic pressure in our body. In the next classes, we will discuss the next important fraction of plasma proteins, that is globulins. Thank you so much.